You are likely familiar with rust, but maybe not the type that's been eating the near-surface steel in the Duluth-Superior Harbor. In parts of this Lake Superior port, corrosion has accelerated to rates resembling those of the ocean. The perforations and pits are most severe in the first 4 feet below the waterline. Many people collaborated to discover that this unusual corrosion is caused by a thin film of iron-oxidizing bacteria. When ice scours these specialized bacteria colonies from the steel surfaces, the exposed areas attract dissolved copper, which pits and weakens the metal. Researchers at the University of Minnesota Duluth have been developing coatings to prolong the life of steel in the Duluth-Superior Harbor and other freshwater harbors that have started to corrode at faster than normal rates.
We've been studying this biocorrosion problem in the Duluth-Superior Harbor now for probably about 10 years. And so now that we have a good picture, our current project is moving on to look at ways we might mitigate this corrosion problem by either preventing bacteria from attaching to some of these steel surfaces or changing the composition of the microbial community such that hopefully we would see some change in the corrosion rate for the better. Cool. Early on, it was a challenging intellectual problem. No one really knew what the cause of this biocorrosion was. And there were some indications that microorganisms might be involved, or either directly or indirectly through their metabolism.
The most significant impact will be to reduce the cost of maintenance of the steel infrastructure in the harbor. Because when the steel heavily corroded, you will have to replace them, and it is very costly. It will cost like 10 million per kilometer of the steel to be replaced, and also recoating them will be another significant cost. So we want to see if our coating are applicable in the industry, then they can use them and then reduce the cost.
So these are the size that actually go in the harbor, and there might be several replicas, and we can put those out here too. So far in laboratory experiments, we've seen using one particular enzyme in a coating that we saw a 50% reduction in the amount of number of corrosion tubercles that had formed, these rusty blisters on the steel. We also saw that there was a decrease of 50% of the area that was covered by these types of corrosion tubercles. And that was our best treatment. We tried several different biochemicals which have been implicated in helping to reduce corrosion. And so those results were very promising from the laboratory. And so now we've moved out into the field, which is much harsher conditions and less controlled. And we're looking at what is the best matrix for the coating, as well as what concentration of enzyme is optimal. And those experiments are still ongoing. And currently we still have test coupons, steel coupons, that are out in the field.